Good morning, and thanks again for joining the 48th Annual JP Morgan Healthcare Conference. I'm Eric Joseph, Senior Biotech Analyst with the firm. Our next presenting company this morning is Black Diamond Therapeutics, and it's my pleasure to welcome company CEO David Epstein to talk to us a little bit about the company. There is a Q&A session after the presentation. Just click the icon, and I'll work in questions where appropriate. So with that, David, thanks so much for sharing some of your time with us this week.
Great. Good morning, everyone, and thank you, Eric. It's a pleasure to be here representing Black Diamond Therapeutics. So I'd like to introduce today's presentation with just our approach, the business thesis for the company, and just remind everybody of the rapidly advancing nature and progress that we've made in the area of precision medicine. So Black Diamond is a precision medicine oncology company focused on the development of master key mutations and master key therapies. The classic approach shown on the left-hand portion of the slide is really dealing with the development of targeted therapies, most prevalent mutations in a single tumor type. It's led to the advancement of the field. It's a $30 billion market, and that has been, I think, great for patient care. With the ongoing expansion of next-gen sequencing and profiling in cancer patients, we've seen also the advancement of the approvals in precision medicine, but unfortunately, less than 15% of patients today diagnosed with a metastatic cancer actually have an available therapy available for them. And this is really the purpose of Black Diamond's MAP approach and the master key approach that we're going to talk about today. We target mutation families to expand the opportunity for precision oncology, shown on the right. And you can see that the prevalence of these mutations is just not located at a single site, but throughout the oncogene. So mutation families actually have the potential to yield significant market opportunities for those cancer patients lacking suitable precision medicines. And I want to spend a moment talking about, what we've accomplished at Black Diamond Therapeutics. So our proof of principle for this master key approach really derives from the development of family-selective inhibitors that are now clinical and late-stage preclinical. And the 4 targets that we have developed these inhibitors are shown in the second bullet point on slide 4. EGFR, HER2, BRAF, and FGFR validated oncogenes, yet there are numerous unannotated and undrugged mutations prevalent in these individual oncogene families. So BDTX 1535 is the latest advancement in our MAP platform pipeline. It's an irreversible EGFR mutant selective yet brain penetrant inhibitor. It's now entering phase 1 for the treatment of patients harboring these EGFR master key mutations in both glioblastoma as well as in non-small cell lung cancer. BDTX 189 is the flagship product for Black Diamond Therapeutics. It's a mutant-selective EGFR and HER2 specific inhibitor that is currently in phase 1 safety expansion studies for the study of this drug in patients expressing master key mutations in both of these families. So the proprietary MAP discovery engine is designed to do the following things. It's designed to predict and to validate novel oncogenic mutant families from population-level genomic data sets, as I think you know. We have pioneered mutant family confirmation-based master key drug design. We'll talk more about that as the talk proceeds. But importantly, this platform provides opportunities both beyond oncology and outside the arena of small molecule drug discovery and development. I'd like to spend a moment talking about the MAP platform. So 3 slides. In essence, we predict the likely oncogenicity of a given mutation, going clockwise left to right. We validate both in preclinical, in vitro, and in vivo models, group mutations into families, and then we employ biochemical and biophysical methods to characterize the structures of these mutant families and thereby develop master key specific inhibitors against these self-same master key families. The next slide I'm sorry, I'm having trouble getting this thing to behave. The next slide actually gives a description at a granular level of the complexity of the mutation landscape for individual oncogenes. So shown on the far left is a population picture of a given oncogene with the mutation lollipop chart shown in maroon. We use computational in silico prediction methods to predict the likely oncogenicity of a given mutation, experimentally validate it, and develop master key mutation families out of it, shown on the far right. The next slide, The next slide shows the ability of our teams to predict the conformational forms of these mutation families. We have deployed a cloud-based computing method to identify regions of the protein that are highly mutated, subject to conformational change, and we can use these methods to determine the effects of these mutations on global conformational change and how they affect the actual active site of the molecule of the targets in question. You can see individual master key mutations drive global conformational change, and master key inhibitors are designed to exploit this vulnerability. I want to spend a moment now talking about BDTX 1535, an oral brain penetrant selective inhibitor of the oncogenic EGFR master key mutations. It is designed to treat patients harboring these oncogenic master key mutations. It is designed to be blood-brain barrier penetrant and to treat patients harboring CNS tumors. It is designed to be selective versus wild-type EGFR and to deliver a favorable safety profile. As we announced yesterday, it is cleared for the phase 1 study and will be dosing patients in the within the first quarter of this year. I'd like to set up the rationale for the study of EGFR in glioblastoma as shown on slide 10. The problem, as we have articulated it, is that patients harboring these mutations in GBM simply do not derive benefit from first-generation EGFR therapies, whereas the self-same drug has shown these self-same drugs have been shown to provide benefit to patients harboring master key mutations in EGFR in non-small cell lung cancer. And I am on slide 10, and I'd like you to focus on panel 1 at the top in GBM and panel 2 at the bottom. You will note that the location of these mutations in glioblastoma is in the extracellular ligand binding domain of EGFR, whereas the activating mutations in non-small cell lung cancer, as most of you know, are located in the kinase domain. So 2 different locations and 2 different behaviors of these mutations. On the far right of slide 10 shows our initial observations that not only are these mutations dissimilarly located, they also have dissimilar pharmacological activity. So in gray is the Ic50 curve shown for EGFR inhibitors such as erlotinib when administered to cells whose proliferation is driven by the expression of EGFR catalytic domain mutations such as the exon 19 deletion. In maroon, you see a completely different Ic50 curve, and one that exhibits a paradoxical activation whereby low doses of an EGFR inhibitor actually activate the proliferation of cells, reaching a maximum activation rate, and then at higher concentrations of these EGFR inhibitors, you actually see inhibition of the enzyme. We have developed data that explains the behavior of these early generation EGFR inhibitors in glioblastoma, and that is shown on slide 11. And simply put, the oncogenic conformation of mutant EGFR in glioblastoma is not the same as what we see in non-small cell lung cancer. And in fact, this mutant-specific conformation is in the form of a locked dimer. It is an irreversible dimer that is formed because of the types of alterations that occur in the extracellular ligand binding domain of EGFR in the glioblastoma setting. This locked dimer is constitutively activated. It has 2 homodimeric active sites. And what we see is that this is a detrimental and potentially unwanted property of this oncogenic isoform. In vivo, we have shown that we can mimic this paradoxical activation with a single dose administered of high concentrations of EGFR inhibitors, such as Tarceva. So high concentrations of EGFR inhibitors like erlotinib will shut down the activity of this kinase in PDX tumors at an initial level. But as the drug dissipates in animals, you see a rebound of activity and then eventually a paradoxical activation where it appears that these EGFR inhibitors are actually driving the dimerization process in preclinical in vitro and in vivo models. Thus, we have concluded that not only do we need a potent and selective and blood-brain barrier penetrant inhibitor, one needs an irreversible inhibitor to avoid paradoxical activation and sustained target inactivation and inhibition. And we will show that data for 1535 on the following slide. I am sorry, but my computer is jumping. Okay, so on slide 12, we show single dose PK/PD experiment on the left, where a single dose of 1535 leads to sustained target and pathway inhibition out to 24 hours. And this results in increased survival, astounding increased survival benefit in intracranial models of PDX tumors associated with glioblastoma, and that data is shown on the far right. We report here an average unbound KPUU. This is the free fraction in rats that is amongst the best in the field, and certainly at least 2-fold better than what is observed for currently approved irreversible inhibitors in the class. This translates then into profound and robust anti-tumor activity across a range of tumor models. And shown here on slide 14 is the data for 1535 in models representing glioblastoma driven by amplified EGFR, models of glioblastoma driven by variants such as variant 2, as well as point mutations associated with EGFR and glioblastoma. We have a range of tumor models that exhibit profound tumor regression in both the non-small cell lung cancer setting. And so we're really excited about the ability of 1535 to promote anti-cancer activity in preclinical models, and excited to be advancing this program into clinical development. This is a list of the preclinical mutation that we have. This is the master key mutation list, and the activity profile for 1535 against all these mutations. These are the mutations that we'll be studying in the clinic. The average IC50 values for these mutations is in the low single-digit nanomolar range, and the selectivity versus wild-type EGFR is in the range of 60 to 130-fold. You'll notice a range of mutations in glioblastoma. It is important to point out that in the field, it has not been commonly recognized that these mutations co-occur within individual patients. And so 1535 solves a number of development problems that have plagued the GBM field. It is now a first-in-class blood-brain barrier penetrant EGFR inhibitor. It targets all forms of master key mutations associated with glioblastoma, and it leads to profound tumor regression without paradoxical activation. On the non-small cell lung cancer side, we are targeting a range of mutations that are essentially undrugged and available for therapy. These are exon 18 mutations shown here, as well as the ability to irreversibly target the C797 wild-type form, as well as the C797S acquired resistant form of EGFR in lung cancer. Slide 15 then shows an example of how we are using our computational tools to model the effects of these mutations on the active site of the molecule. You'll note that the mutations in GBM occur distal to the active site, yet other mutations in EGFR are associated with regions around the ATP binding site. And this data just simply exemplifies the complexity of the mutation landscape and the ability of these mutations to impart subtle yet profound structural changes in the ATP binding site. And here we show the ability and the need to target both C797 as well as the acquired mutation C797S. I'd like to end I'd like to now show an outline of the phase 1 clinical trial design. This is a biomarker-driven first-in-human study that, again, just cleared IND allowance yesterday. And so we're really proud of this. We'll be simply running a dose escalation study, single patient dose escalation cohorts to bring us to, I have to apologize. I'm having trouble with my computer this morning. So single patient dose escalation study, Boyne design, where at appropriate doses, we'll be able to move into multi-patient dose escalation cohorts. We'll study the drug in 3 different settings: GBM patients harboring EGFR mutations associated with GBM that we've previously described, non-small cell lung cancer patients harboring intrinsic resistance mutations in exon 18, as well as non-small cell lung cancer patients harboring acquired resistance mutations. We anticipate reporting on data in the second half of 2023, but more importantly, I want to come to slide 17 and just comment on addressable populations. We see this as an area of high unmet need. Our entry into the GBM space will be in recurrent GBM, where the population is approximately 25,000. We have a direct opportunity to go into frontline setting and the ability to treat patients with these mutations in the presence of in combination with temozolomide. Total addressable population in the GBM space is around 61,000. Of course, I think many of you are quite familiar with the lung cancer space. Our entry point into this is both in the intrinsic resistant family, exon 18 mutation family, approximately 12,000 addressable patients, as well as in the classical acquired resistant mutation family, approximately 9,000 addressable patients. A drug such as we've described will have the ability to move upwards in lines of therapy such that approximately 160,000 patients in the 4 major markets can be addressed by a drug such as 1535. I want to spend a few moments speaking about BDTX 189. As you know, this program has been in phase 1 and is still in safety expansion studies. FDA endorsed the recommended phase 2 dose for this target at 800 milligrams QD non-fasting. These levels and the recommended phase 2 dose, the QD dosing regimen, and the PK that we observed were in line with preclinical expectations. We showed that BDTX 189 was well tolerated with minimal evidence of systemic wild-type EGFR-related adverse events. We demonstrated early signs of anti-tumor activity in 2 patient cohorts, EGFR exon 20 in lung, as well as HER2 amplified solid tumors. And as we have indicated, we will continue enrollment into the safety expansion cohort to gate the next steps in our clinical development strategy. I want to go to slide 19 and again reemphasize that the FDA has endorsed the 800 milligram QD non-fasting as a recommended phase 2 dose. And I simply want to point out that this is well tolerated. No DLTs have been observed at the recommended phase 2 dose. And we will use the assessment of this drug in the safety expansion cohort to gate future development of this as a single agent or as an agent to be used in combination. Slide 20 is a summary of the safety profile. And again, on the left, I think many of you have seen this. BDTX 189 was designed as a wild-type sparing EGFR mutation selective inhibitor. potential best-in-class with respect to the preclinical profile, and potential best-in-class, as you can see on the right. What we show here is the 800 QD non-fasting cohort, N equals 15. I do want to point out on the previous slide that we studied this drug, we have studied this drug in upwards of 59 patients, and today we are summarizing the non-fasting cohort at N equals 15. Essentially, all of the toxicities are medically manageable, grade 1 diarrhea in approximately 51% of the patients. Nobody has required prophylactic antidiarrheals or antiemetics. A low grade 1 vomiting, essentially little to no rash. So the conclusion that we have limited toxicities associated with systemically available EGFR is our thesis, and in line with expectations from our preclinical studies. And then lastly, I want to just comment again on the several patients that we have seen in the study that have remained on study, and this one today is our lung cancer patient with an EGFR exon 20 deletion, still on study out to 10 months, target lesion progress over time. And just important to point out, the lung at the start of the study in this heavily pretreated patient really essentially looked like a snowstorm for 6 weeks, clearing of the lung, off oxygen, and continuing to do well. So BDTX 189 can provide benefit in this clinical setting. I want to end the presentation then with a quick discussion of our oral brain penetrant inhibitor of oncogenic BRAF mutations. This agent was designed to be potent and selective for all three classes of BRAF mutation, class 1, class 2, as well as class 3. It has been designed to be brain penetrant. We have achieved that, and we'll show that data in a moment. It has been designed to inhibit dimerized RAF kinases, any mutation background, and to avoid paradoxical activation. And we'll just point out that paradoxical activation is a critical feature. It leads to cutaneous toxicities. It actually ameliorates and abrogates the activity of inhibitors that are otherwise appropriate for clinical development. So it is an essential feature of our target product profile. We're guiding today that we'll be initiating IND-enabling studies within the year as we begin to finalize our characterization of this BRAF compound that I'll show you momentarily. So shown here is a summary of the in vivo data for our BRAF lead, master key mutation lead. So it shows profound tumor regression in models of class 1 as well as class 2 or class 3 mutations in BRAF in comparison to clinical agents which simply can't target the class 2 and class 3, or simply what we believe is the key competitor in this field for us, and that's belvarafenib, shown here at its clinically relevant in vivo dose at 15 mg/kg. Low doses of the BDTX BRAF compound in all three models leads to profound tumor regression. But more importantly, we have shown in an intracranial tumor model driven by BRAF V600E mutation, the ability to sustain inhibition at both doses days after administering. So shown here is belvarafenib at day 0 and day 15. And you can see belvarafenib, though an active agent, still exhibits the tumor growth in the intracranial setting. We see no evidence of tumor growth at the low dose as well as at 10 mg/kg. And these data lead to sustained life and sustained survival in this intracranial model as shown on the far right. So we're really excited about this program. And the reason we're excited about it is that this particular program exemplifies not only the ability to grow the commercial landscape of our products, through the MAP engine deorphaning of rare and recurrent driver mutations, validation of these mutations, and then assess them, testing them in our preclinical models. So this is an approach that is unique to Black Diamond, as it provides the ability to grow the addressable patient population by deorphaning rare and recurrent overlooked mutations. The total addressable population for the BRAF compound is outstanding, and we are looking forward to providing benefit in this large group of patients. It is important to point out that BRAF mutations in melanoma and other solid tumors such as lung cancer have a high degree of prevalence of metastasis to the CNS compartment. And so entry into metastatic lesions in the brain is one path that the company has, as well as an early entry into the treatment of class 2 and class 3 mutations in various solid tumors. Approximately 190,000 to almost 200,000 patients available for treatment with our BRAF drug. So I'd like to take the opportunity just to thank the team. All of this work has been done by my co-founder and her team, Liz Buck, a great team of collaborators with Karsten and Fang. And I want to take the opportunity just to remind everybody on the following slide of the milestones that we will be working towards over the coming year. Clinical data for the 1535 program in the second half of 2023, but of course, any opportunity to report on the progress of this program throughout 2022, we will take. We will report on the safety expansion data and provide an update throughout 2022. We are on track and on time to initiate IND-enabling studies in 2022 for our BRAF program, and we are continuing to move forward with our early pipeline and our confirmationally based drug discovery approach and development candidate selection in the late 2022 for FGFR or other undisclosed programs. And with that, I'd like to end here and thank you for your attention, and my apologies for the computer issues that I was facing this morning. So thank you very much.
Thanks for that, David. A couple of questions coming in through the portal, so I'm going to start there. First is on BDTX 1535, asking you to comment on the biochemical selectivity of the compound for the spectrum of mutations that you highlighted that's designed against versus wild type, right? You wild type EGFR, right? And I think they're perhaps referring to inhibition and dissociation coefficients, not necessarily just the in vitro cell assay data. And sort of what sort of lessons from the 189 development sort of have been incorporated in the selection of that compound?
So these are still master key approaches. We think that the MAP platform and the grouping of mutations into druggable families is a meaningful scientific thesis, valid scientific thesis, and commercially relevant and important for patients. So this is a spectrum-specific drug, meaning that it has high selectivity for EGFR. It does not target HER2. So what you see in the bar graphs, and this data, of course, is on the website, are the Ic50 curves, and Ic50 curves in cell lines that are driven by these particular oncogenic lesions. So we are showing low nanomolar Ic50 values. We essentially are developing our drugs in whole cell assays, targeting the full length protein. In the GBM setting, I will point out that if the question is this a potent biochemical? Is it potent biochemically? Absolutely. But you can't study GBM mutations in a biochemical assay. These are mutations that are on the outside of the cell. So We think that the learnings from 189 are in the following categories. We developed 189 as a rapid on, rapid elimination, so-called hit and run drug. We developed 1535 with the opposite in mind. So 1535 has a projected human half-life of approximately 15 hours. It has extended human blood stability. And we believe that this will favor dosing and exposure at lower levels than what we achieved with BDTX 189. What I mean by lower drug levels. Obviously, we have to go into the clinic to see how this plays out, but these are two fundamentally different candidates. This is an irreversible drug, just like BDTX 189. 189 is not blood-brain barrier penetrant, yet it is irreversible. It targets the C797S lesion. 1535 not only is blood-brain barrier penetrant and irreversible, its warhead targets both wild-type cysteine as well as serine at the C797 position. And while I didn't go through that data in the presentation, the demonstration of the irreversible nature of 1535 with blood-brain barrier penetrance data is all available on the corporate website.
Okay, great.
Very different drugs.
Okay, that's helpful. And just with respect to the phase 1 study, as you laid out there, I guess how should we be thinking of total number of patients that you could enroll? Is there any ceiling to the number of patients that you could accrue? Appreciating that you are kind of going through a fairly rapid Boyne escalation scheme, and do you want to arriving at a phase 2 recommended dose? Is it important to see? Is important to track biomarkers in both target populations here, both in GBM and in lung cancer patients?
So I think it will be important to track biomarkers of activity, and we have in this phase 1 study design the ability to follow the activity both in the CSF compartment as well as in the peripheral compartment. That's been designed into the study. I think the other important point is that we will be studying the drug in single patient dose escalation cohorts until we reach a certain level. We'll then shift over to the multi-patient dose escalation cohorts. We're not giving guidance as to the number of patients in the study at this point in time, but I think it's important for everyone to understand that this is a study where every patient who is enrolled is going to be biomarker positive. We're going to be studying the drug at the relevant, in the relevant patients with the relevant mutations, either GBM patients and attendant mutations, lung cancer and the exon 18 mutations, or lung cancer carrying the C797S lesions in EGFR. These will be 3 distinct cohorts of patients that will be assessing the drug from the get-go. So we expect to see early signs of clinical activity in the multi-patient dose escalation cohorts, and it is there that we'll expand out and use this method to getting to the recommended phase 2 dose. So a version of essentially an approach to clinical development that is quite different from what we did with BDTX 189, where here we're just looking at 2 different tumor types, a narrow set of master key mutations, and of course, and the ability to study these patients at pre-recommended phase 2 dose levels.
Okay. The biomarker analyses that you're doing, particularly in the GBM patients, can you just sort of unpack how that process works? Is it serial biopsies that you need to look at, and EGFR pathway inhibition? And just curious about how that works out.
For enrollment criteria, every patient will have a solid tumor sample because every patient in the GBM setting undergoes surgery, radiation, and then temozolomide treatment. And so we will have primary diagnostic specimens. In fact, it's mandatory. And EGFR mutations and amplification proof will be mandatory for entry into the clinical study.
Okay. That's for entry. But in terms of PD analysis on drug, what informs? What biomarkers, I guess, are you? Or are there biomarkers? Is the primary idea just looking at, after establishing safety, you're looking at tumor responses at that point as evidence of activity?
Yeah, so there's the ability to look at the study of the drug in a so-called leptomeningeal cohort. That's part of the phase 1 study design. We have the ability to open that cohort at doses that approximate what we believe is the recommended phase 2 dose in the GBM setting. And there, one can study circulating tumor DNA, one can study circulating tumor cells, and get a sense of target engagement and literally the PD activity of 1535. We can also do PK analysis in the CSF. So we think that there's going to be a wealth of data coming out of the GBM assessment. Okay,
great. And then on 189, where you're expecting to provide an update this year, I guess how should we be thinking about additional patients coming into the safety expansion cohort? And just picking up on a question that's submitted here, the decision as you lay out, or next steps are going to be informed by safety in that trial. I guess the question is, why not efficacy?
Safety expansion just means it's part of the phase 1 study. It's of course we'll be looking for activity. And if the question is around what are the deciding features for gating forward, we're just not in a position to provide guidance at this point in time. But obviously, this is a study where we will be looking for signs of activity that are meaningful and that can provide patient benefit in what I think all of us will admit is a very competitive space. Now, I do want to remind everyone 189 is developed to target a range of mutations in a range of tumor types. I think we and investors expected that an early sign of clinical activity would come out of the lung cancer exon 20 group. But the investment thesis there, I think, is challenging given that post-ASCO of this year, there have been 2 drug approvals. In the area, one small molecule led by Takeda, one large molecule, the bispecific amivantamab, developed by Johnson & Johnson. And so, that is complicating. We have one additional drug under review now with breakthrough designation. And so getting a proof of concept data in that setting, I think, is what the team needs to look at. We are a science-based company. We're going to follow the science. But realistically, the competitive landscape in one area of BDTX 189 is challenging, and I think we just want to recognize that challenge. And we are going to follow the science. We'll see what the efficacy data looks like in the safety expansion cohort. We'll inform the investors of that. But this is an opportunity, and I think we didn't get asked this question and didn't come up in the presentation. This is an opportunity to extend the runway. We're ending the year at $210 million in cash. We are now giving guidance that the runway will take us into 2024. We can focus our resources on 1535. We can focus our resources on early stage pipeline. We've made great advances in the development of the MAP platform, particularly in the arena of conformationally based drug design. I think you saw elements of that with the 1535 program. More of that will roll out with what we're doing with BRAF. Clearly, we've developed expertise in the ability to deliver drugs that can cross the blood-brain barrier at high levels. So the team, I think, I'm really proud of what they've done. Team has remained, I think, committed and focused. And I will point out that the 1535 IND guidance was for the first half of this year. We obviously submitted in the end of 2021. got approval on the first day of JPM. So thank you for that. It must have been a blessing that we had the meeting, and we're excited about where we're going and what we've been able to accomplish. So thank you for that.
Okay, great. Well, I think we'll leave it there for time. David, thanks so much for sharing some of your time with us this week, and thanks everybody for tuning into the session.
Yeah, thank you, Eric. Thank you, everybody.